Okay, so good afternoon, everybody. I was told that I can start. So first of all, welcome back to the second day of Mathematical Models in Epidemiology lecture. I'm going to share my screen. And I hope you all can see it. So after the theoretical lecture from yesterday, where we have learned how to develop a mathematical or an epidemiological model, how to do the stability analysis, and how to interpret the results, we now go for the fun part, where we deal with real-world problems. I have two case studies to present. The first one would be on dengue fever today, and then on Thursday, we present our work done on COVID-19, applying all the concepts that we have seen yesterday. So the lecture today would be to talk about the impact of the modeling and the data analysis within public health problems. Again, my name is Maíra, Aguiar, and if you need any help, please feel free to contact me by email. You have my email in the first slides from yesterday. Okay, so before we start, I give you a brief of introduction about dengue fever epidemiology. I'm not sure if you are familiar with this infectious disease, but this one of the most important infectious disease in the world. It is transmitted by a mosquito, and after malaria, this is the most social and economical problem in terms of public health worldwide. It is a leading cause of illness and death in the tropics and subtropics, and we have more than 1/3 of the world population living at risk of acquiring dengue infection, as we can see here in this map. So the colors have shown where are the most risky areas to live, where the virus and the mosquito transmitted disease are present worldwide. You can see that it's no longer restricted to the tropical areas, although mostly there, but we have seen cases also occurring in Europe. We don't see here because it's up there, especially in Portugal, Spain, and France. So dengue is a very interesting disease because it's not like measles, for example, that we have a single virus transmitting, and then after you get immune, you simply do not get infected anymore. Or there is a very good vaccine against measles that after receiving the full immunization schedule, you are immune and you are protected against this infection. So dengue is special, first of all, because there are 4 antigenically different viruses that can cause the disease. And it's special because these 4 antigenic viruses are closely related, which makes some confusion after a primary infection, leading to eventually a more severe illness when the host acquiring a secondary infection. So those 4 antigenically distinct viruses are called dengue 1, 2, 3, and 4. And in this map here, you can see how fast those serotypes has spread around the world. This is the presence, the global distribution of the serotypes in 1970s and up to date. And you see that it was very fast. So the 4 serotypes are now co-circulating in many parts of the world. This map here also highlights the importance of having data on Southeast Asia, where the 4 serotypes are coexisting at least since 1970s, which gives us some more information in terms of coexisting, co-infections eventually, and secondary, tertiary, quaternary infections. So we need to understand the biology of the virus. We need to understand the epidemiology of the infections, and some important key aspects that we need to consider when developing a model for this specific disease is that infection by one virus, called here serotype, will confer lifelong immunity to that serotype. So once you get infected, for example, with dengue 1, you are fully immunized and you cannot get a secondary infection with the same virus. You have also a period of cross-immunity that's called temporary cross-immunity period that protects you against infection during a restricted period of time. In the literature, it lasts between 3 to 9 months. So in summary, if you get infected with dengue 1 first, you are lifelong immunized against this serotype, and you have a period between 3 to 9 months where you cannot be infected with any other of the dengue viruses. However, after this period of time, this protection decays and you are considered susceptible again, being able to get the secondary infection with a different serotype. So dengue has a wide spectrum of clinical presentation. Most of the primary infections are asymptomatic, but some of them can become severe cases of the disease, which is correlated with a secondary infection, and I will explain to you why. So most of the patients recover from a primary dengue infection because it's just like a flu illness. Whenever you have any symptoms, most of them are asymptomatic, and a small proportion would then progress to severe disease, which is characterized by hemorrhagic symptoms. As I have mentioned, epidemiological studies have associated severe disease with secondary infection due to a process described in the early 60s called antibody-dependent enhancement that I will try to explain here in this scheme. So this scheme here shows what are the immunological response in the primary dengue infection with a serotype 1 and a secondary infection with a different serotype. So what happened is that when we have a pathogen invading a human host, this pathogen is invading cells. In the case of a virus, it's invading cells, is replicating to the cells, and the immune response are mediated by antibodies first, where we generate the so-called non-specific antibodies or IgM antibodies, which is able to bind to this virus and to clear the infection. While this IgM is doing the work here, we also produce the so-called memory antibodies or IgG, which is the antibody which will confer the lifelong immunity to that specific serotype. So a parenthesis here to say that we do not have asymmetry between serotypes at this moment. So this label 1 and 2 is just to differentiate that the first one is different from the second one, okay? Okay, so after the primary infection, you enter in the period of time called temporary cross immunity, where it is speculated that you have such a huge amount of antibodies circulating in your organism that you are protected against any related serotype infection occurring during this period of time. However, the time passes and these levels drop to a certain level that the individual is already susceptible again. Okay, so after this period of temporary cross immunity, the individual becomes susceptible. You can then acquire a secondary infection. If a secondary infection is via the same serotype as the primary infection, this pre-existing memory antibodies, this pre-existing IgG will increase, and notice that this reaction of secondary infection occurs in most of infectious disease that have that we can model with the SIR type model, where individuals become recovered and then susceptible again, such as influenza, for example. As close as the viral is, as close as the virus of the secondary infection is from the primary infection, more immunity you have, more protection you have. So with dengue, it's not different. So by being infected with the same virus, you have a pre-existing antibodies being activated, able to bind to this virus and to clear the infection. So normally, secondary infection with the same serotype are not detected at all because you have immediately a clearance of infection, and the individuals are supposed not even to have enough virus to transmit. So they, in theory, they do not count into the force of infection of dengue epidemiological dynamics. Okay, but we are interested in knowing what happened when you get a secondary infection with a different serotype. Remember that I have told you that the dengue serotypes are antigenically very closely related. And what happened is that the different dengue virus enter in the cell, and instead of being recognized as a different virus and starting this process again, it is recognized as being the same virus. So the immunological process starts very fast using the pre-existing antibody, which is specific for the primary infection virus, but it is able to bind into the new virus. However, due to this specificity, this antibody is not able to neutralize the virus. So you end up with a complex antibody virus with this virus still activated and helping this virus to enter in the susceptible cells and enhancing the replication of the cells. So what you have is an enhancement of viral replication due to this complex of inactivated virus and pre-existing antibody, which later on will turn on again this primary response with IgM, and only then production of specific antibody against this virus. The thing is that when we have this cascade of enhancement immunological responses, you also have an enhancement of symptoms, and most of them are described as blood leakage or hemorrhagic symptoms. And that's why a secondary dengue infection with a different serotype mediated by pre-existing antibodies will probably lead to a severe disease with hemorrhagic symptoms. So what we know up to now is that the infection between the primary infection and the secondary infection do not occur at space of less than 6 months. And that is not only because of the temporary cross immunity we have discussed here, but also because dengue is a seasonal disease, which means that you have a high transmission after rain because you need the mosquitoes to replicate. And you have low transmission in the winter where the mosquitoes cannot eventually survive for a long time and transmit the disease. There is no specific treatment for dengue, and all the severe cases will require hospitalization once the individuals have some hemorrhagic symptoms. Okay, till end of 2015, 2016, there was no vaccine available for dengue. Up to date, we have one licensed dengue vaccine, which is developed by Sanofi Pasteur. And I have a second vaccine, which has just finished the phase 3 trial, developed by Takeda Pharmaceuticals. And we are going to discuss the results of this vaccine later during the day. Okay, this is only to illustrate that this only licensed dengue vaccine called Dengvaxia have shown difference in efficacies by age groups, and we try to understand why that happens. For example, just to illustrate here, this is the vaccine efficacy for the first 4 years after the trial started, where we see the vaccine efficacy is in its positive efficacy, which means that the vaccine is protecting against disease during the first 2 years, but drop to the negative vaccine efficacy. That means that vaccine was causing more cases than into individuals which never received the vaccine. For younger than 9 years old of age, and for older than 9 years of age, you see that the vaccine efficacy has changed, but still were above or were having some positive impact. So regarding the new vaccine, the Takeda one called Dengvax, the trials were just published recently. And here we can see that the results for the overall vaccine efficacy after the 12 months and 18 months of the trial, we see that overall vaccine efficacy was around 80%. And as the time passes, it starts to decrease. This is also observed in many of the vaccines we have up to date. We have to observe to see if it doesn't go even lower. A vaccine has to be a minimum of efficacy in order to be licensed and implemented. And for the Takeda vaccine, the situation is that as time passes, the vaccine efficacy is decreasing. And therefore, we need to look at closer and to monitor this, what's so-called phase 4 of a vaccine trial, how it goes. What we observed also in the vaccine efficacy of Takeda is that there is difference between individuals vaccinated as seronegative, so naive individuals which have never been exposed with dengue, from individuals which have been already exposed, or the so-called seropositive individuals. We see that the vaccine efficacy for seronegative individuals is smaller than vaccine efficacy for seropositive individuals. Here we can see the difference per serotype, and we see that we have 2 different vaccine efficacies. Okay, so here I just compare the Dengvaxia from Sanofi and the Takeda vaccine efficacy, and while in the Dengvaxia you have not similar vaccine efficacy for all serotypes, you have here dengue 2 and dengue 1 with a lower vaccine efficacy, as opposed to dengue 4 and dengue 3 with a intermediate to good vaccine efficacy. And in the Takeda, we also see this difference between serotype, where we have dengue 3 and dengue 1 with a low vaccine efficacy, dengue 2 with a good vaccine efficacy, and that can be explained because the vaccine is based on the backbone of this dengue 2 virus. And the dengue 4, the result is not really clear to us yet. But we are going to discuss that when we implement that into the model, just to you to have an idea that we have information about the biology of the disease. We can develop a model, we can extend the model in order to analyze the controls we have at hand, in the case vaccine or vector control. And from that on, you have to evaluate the data which is available and then implement it into the model to see what are the effect of these vaccines. Okay, for the case of dengue, we have plenty of data and plenty in different types of data. And of course, the data you have at hand and you want to work regarding the question you want to answer will define the model framework you are going to develop. So just to illustrate here, we can use data on serotype. So this is for Puerto Rico, for example, the dynamics of dengue 1, 2, 3, and 4 over the time. We have data on mosquito eggs when you want to answer any question about controlling vectors in a given region. You have data on incidences, and here's, for example, for Thailand. This data about incidence can be also spatially distributed. This is the map of Thailand with the 77 provinces, and we would have this type of data here for each one of the provinces. And we also have data on antibodies, on viral load that allows you to develop within the so-called within host model dynamics. Okay, so the first question is, can we describe what happened within host using a simple model? I mean, yesterday we have seen the SIS, we have seen the SIR, we have seen the SHAR model. So what is the complexity of a modeling framework you need to describe the dynamics of, for example, a within host immunological response for dengue? And the answer is yes. You can develop a modeling framework to describe the antibody-dependent enhancement process, for example, in the secondary dengue infection with a different serotype. You can evaluate the role of temporary cross-immunity in the immunopathogenesis of the severe disease. For example, what happens when you get a primary infection in January and a secondary infection after a year, or a secondary infection after 10 years? Are you going to develop a more severe disease or a less severe disease? And we can also evaluate the impact of these imperfect vaccines that I just commented to you on the disease prevention and control. So this is a work for my PhD student. She's working, Vista, she's here with us. She's working on the within host dynamics for dengue. And I will try to give a bit of flavor of what we have done so far, focused on within host modeling first. So you remember here this scheme, and we are going to analyze how is the dynamics of dengue recurrent infections, including this pre-existing antibody here after primary infection and in a secondary infection with the same virus and with a different virus. Okay, so we start modeling dengue primary infection with a serotype type 1, and we see here that we have the human host being infected with the virus. This virus invades the cell, replicates, and the immunological responses start by generating this unspecific antibody IgM, which is able to bind into the virus and clear the infection, while this lifelong immunity antibody IgG is also produced, and together they lead to a temporary cross-immunity period. And we can write down that in terms of a model, so we can just describe that with a model, where we have susceptible cells meeting the free virus, becoming infected with an infection rate A. So these target cells are specific ones, and they are called dendritic cells and monocytes. We have a third cell, which is called macrophage, and this cell is very important for the immunological response because this is the cell which is going to phagocyte this virus and will transform, also would be differentiated in the so-called presenting cell. This presenting cell is basically this one here, which is going to present the virus and will trigger the production of those antibodies. Okay, presenting cells will trigger the production of antibodies IgM, which is in a specific with production rate alpha M. And later on, antibody IgG, that is the specific one against this virus with a production rate alpha G. So what happened is that now the free virus will be bound to this unspecific antibody IgM with this rate here, and will generate complexes. So those are the complexes here, IgM dengue virus. And after the IgG being produced, we have this free virus also bound to this specific antibody G, generating complex IgG. Sorry. Here we do not see it, but it would be the same, this virus here bound to this antibody here, and this would be recognized by the so-called killer cell and would clear the infection. Okay, so after the infection is cleared, this complexes just vanish because they are proteins and they are disintegrated and you are clear from the infection. Okay, so we can evaluate this type of modeling in blocks where we check the viral entrance and viral replication, then the differentiation of the target cell into a presenting cell, and then the triggering the antibody. So we have 3 blocks to describe a primary dengue infection with a given serotype. These 3 equations here shows basically that we have a constant input of target cells, and in this case would be macrophages and dendritic cells. These macrophages have a natural mortality, and they decrease by being infected with the virus, by meeting the virus, okay? They become then infected cells with the same rate A as B. And these cells have not only the natural death rate, but also the death by infection, because after they infect, the virus starts to replicate and they explode, releasing more virus with this rate, with this factor kappa here. So we assume that a certain amount of virus goes out of the cell after the cell is dead, and this virus would decrease with a reduction rate B when infect new cells again. Okay, and we can look at the viral production. Here we have the same graphic, so the natural scale and log scale, and we can see that for this model without any immunological response, so only the viral replication, you have an exponential growth of the virus until it's saturated. And of course, without any immunological response, the human host would die because we just get all cells infected and you cannot survive with that. So what we observed first here is that the viral load increased exponentially at the beginning after day 2, and it goes to a saturation when we look at the log plot here. Okay, so but we have immunological, good luck, we have immune system and we can fight this infection. And what happened is that this special cell here, which is also target cell, but now called macrophages, they are produced also at a constant rate. They have also a natural death, but they get infected with the free virus, which is around here. So they phagocyte this virus and they differentiate into presenting cells. And these presenting cells is then showing up the antigen of the virus, which triggers the production of the first antibody called IgM. So the first antibody IgM is produced conditioned to the presentation of the antigen by the presenting cells. They decrease by binding into the free virus and also in its natural mortality rate. So in summary, you have monocytes becoming presenting cells, decreasing the viral load by antigen presentation and antibody production that neutralize the virus in the case of primary infection, first by IgM, and then we have the, and then we have the production of IgG that we are going to say, we are going to see soon. All right, so the second block here is represented by those figures where we see the viral load, so the exponential growth of the viral load to reach its peak and then decreasing at day 6, more or less. And that is the production of the antibody IgM that after, so remember that the virus starts to appear after day 2 after the infection. And the IgM appears, well, the free IgMs only appears later on, but that is because when they start to be produced, they immediately bind into the virus, and we no longer have free IgM, but only complex. That's why we cannot see it here. We are going to see it in the next slides. So when putting the viral load and the IgM together, we see that we have a peak of the viral load decreasing with the increase of free IgM, which afterwards just remain the IgM after the viral load is cleared. So the IgM is used to neutralize the free virus. So if you would plot not only free IgM, but also the complex IgM virus, you would have another curve here showing that the IgM is acting binding to the virus, and that is causing the clearance of the infection. Okay, so what we can observe here is as well is that how long this IgM lasts in our body. And so here's the viral load that we see that is resolved less than 10 days. The IgM start increasing, reach its peak at day 50, and after 90 days, accordingly to what the immunological literature shows us, it decays to a certain level that cannot be detected anymore. And this is only to illustrate how it would look like the viral load and the antibody production over a period of a year during the infection and over a period of a year. So we see that the IgM decays over time, and at day 90, according to the literature, this IgM is no longer observed or detected by antibody tests. And we see that, in fact, the model can represent well this decay of the levels of this specific antibody. Okay, so we can look at it very closely and we see what is the behavior of this IgM. Sorry, the colors here are shifted, but so here we have the viral load, and here we have the IgM alone, and we see that we have a hump here occurring immediately. So let's assume that the virus is introduced here. We see its exponential increase after day 2, but when you look at the IgM, we see that immediately after the introduction of the virus in the human host, you have a hump of free IgM, which then disappears and only appears again after the resolution of the disease, after day 6. And we were wondering what was that. So we figured out that we are looking only at the free IgMs, and all the IgMs are used here in order to fight this period of high viral load. So then, in order to see anything here, we would need to plot the complex virus IgM and not only the free IgM. Of course, after the viremic period drops and the free virus disappears, you will end up only with the free IgM, so not with the complex anymore. So just to see here how it looks like together. And we were wondering what happened here. We have such amount of virus and no IgM. No, that's not it. We have the IgMs, but the IgMs are all in use, and we have complexes here. And we see how it looks like. Well, I can even show you now from the first slide. So this red curve here is where we plot the free virus and the complex IgM and the complex IgG going on, and you see here is the viral load, and here is what we see as a free IgM in green. What we see is a complex IgM, so it's much higher than free IgM, and that's why we do not see by plotting only the free IgM alone in the graphics. Okay, so the second question would be, can we explain why do we need to have a specific sample to test if the individual is positive or negative for dengue infection? So what they say is that once you have symptoms, you have a period of time that you have to collect the blood to test if it is positive for this specific antibody for dengue, IgM or IgG. And that cannot be done during the period of time that you have a higher viral load, because you have no free IgM available, you have only complexes, and you no longer can get the positive results for this period of time. Okay, so and that can be answered because of this hump here. So if you test individuals for looking for a free IgM in this period of time, you are going to find a positive IgM individual. But if you look at here, you cannot find, because all the IgMs are in use to fight the new virus. So in this period of time here, you can rather look for the virus, but not for the IgM specific. And then you have a period of time that you can try to find IgM here, up to these 90 days where the levels are such as the immunoassay, so the the diagnostic test can get the free IgMs available. Okay, so the system is then finished when we add these complexes, which are created by antibody dengue virus, IgM, and IgG, which then goes to 0 after the infection is clear, because those are, as I said, proteins, and they get disintegrated over time. And here to see the full process again, where we see the immunological response mediated by IgM appearing first, and then by IgG second. Here we have the viral load, IgM and IgG. And here in the longer scale that we have the viral load, IgM dropping at day 90 to a certain level, while IgG is in a smaller concentration, but staying for longer time. So that's the so-called lifelong immunity antibody. And this is very interesting scenario that we could describe very well what is known and what is available in the empirical immunological literature. We do not have data for that yet, but I mean, qualitatively, the results are really promising. So for the primary infection, then we can plot all together. We have the free virus here. We have the IgM in green. We have the IgG in purple. And we have the complexes in blue. So what happened in subsequent infections? As I showed to you the scheme already, you have a possibility to have a primary infection followed by a secondary infection from the same serotype. You have an activation of this pre-existing antibody. This pre-existing antibody is able to bind into this virus, and because it is specific to this virus, can clear the infection immediately, and you have no production of enhancement of viral replication. And so we use the same model as I described for the primary infection. The only thing we have added here is that for the IgG, we have an activation of the pre-existing antibodies, which makes the IgG to appear first, then the IgM. The rest is the same. And when we look at it, we compare the primary infection with the secondary infection, and also we got a very interesting result in terms of immunological literature and in terms of what to expect when we get reinfected with the same virus. So for the primary infection, you remember you have a curve here for the viral replication. You have first IgM appearing and then followed by the IgG. The IgM is supposed to drop after 90 days and the IgG is supposed to continue. With the same virus, you have first the IgG appearing, so the activation of this pre-existing IgG, and only then you have the production of IgM. And because of that, because of the specificity of the pre-existing IgG, you have a very low viral replication, and the viral load is much smaller than in the primary infection. This is also observed in the literature. It's so difficult to give lectures online. I mean, if you have any question, please feel free to open up the microphone and ask, okay? I cannot see you, and I cannot see any question in the chat. So I will continue as it goes. Please let me know if you need help. All right. So what happens if you have a secondary infection with a different serotype? Again, our scheme here, and we see that now the secondary infection is generated by a different serotype than in the primary infection. You have the similar behavior. We have an activation of the pre-existing antibody, which is able to bind to this new virus, but it's not able to neutralize this virus as it happened in the scheme before. So this complex IgG new virus helps the virus to enter in the cells because it is recognized as a complex that should be killed, should be phagocyted, but the virus is still activated. So instead of being killed, these viruses are replicating more and more and more. And the human host experiences a higher or enhancement viral replication, which triggers the enhanced immune response starting again from the beginning. So they start to create this IgM, which is in a much higher level, and the production of this IgM is harmful to the body. So it leads also to a severe symptoms, mostly showing some hemorrhagic symptoms. And if this guy survives, only then they are generating the specific IgG or memory antibody to this specific virus. Okay, so here we go. The same model as the primary infection, the same model as a secondary infection with the activation of the pre-existing antibody, but now we have a term here where this antibody virus complex is no longer clearing the infection as we saw in the secondary infection with the same serotype, but it's helping the infection to be enhanced. So you have this complex, unspecific IgG binding to the virus, infecting susceptible cells, which will become infected cells and would increase the viral replication. And when you look at it, we see here that you have a viral, an increased viral load. I'm going to compare all the graphics in the next slide. We have the same process with IgG appearing first, then IgM. We have an enhanced immunological response mediated by these complexes, antibodies, viruses. So here are the three scenarios: primary infection, secondary infection with the same virus, and secondary infection with a different virus. We have the viral load in the primary infection, first IgM and then IgG. In the secondary infection with the same virus, we have a lower viral load, first IgG followed by the IgM. And in the secondary infection with a different serotype, we have a much higher viral load, which is correlated to symptoms. And that's why this scenario here is correlated to severe form of the disease. The first immunological response mediated by IgG, it doesn't work. And then you have a production of IgM leading to a much more enhanced immunological response, which would lead to severe symptoms. Okay, so that's so far we go. We have a very good qualitative result between the model and what we have in the literature. And we are now in the process of getting data in order to parameterize the concentrations of the viral loads and the antibodies during the primary, secondary infection with homologous serotype, so it means same serotype, and secondary infection with heterologous serotype or different serotype. So this is the first experiment and the first modeling type using the data on immunological aspects of a given disease, in this case, dengue fever. Do you have any questions here or suggestion? So, no? Shall we continue? Let me see the time. Okay. All right. So I will continue then. Now, as I showed to you, we have several types of data. This is one type of data that allowed us to develop the so-called within host model. And my question is, when I have incidence data, so number of cases, and in the case of dengue, should be symptom cases, because the asymptomatic, we can never have this type of data unless we are following a cohort and following that population and testing them every day. So the most common data we have is data on people which have been admitted to a hospital and then tested, and then they are counted as a symptomatic dengue. How can we model this type of data? Can we describe the disease transmission or the dengue infection using a simple SIR model that we have discussed yesterday? So this to illustrate how much complexity do we need. First question we ask, how much complexity we need in order to generate a model that gives this type of output. So here we have in blue a modeling output in terms of time series, and we have in red the data. And we see here that the data is quite irregular, and although the model cannot match this 30 years of data, it can resemble very well the patterns of high outbreaks followed by small outbreaks and then high outbreaks and small outbreaks, that you see that in the end, it's a chaotic scenario, but we are going to get there. Okay, question. How much complexity we need to get this type of behavior? can we describe the oscillations observed in the data? You remember yesterday, I have shown the SIR model with seasonality that is much more complicated, giving already chaotic dynamic as soon as we add seasonality into the system. All right, so let's see what we need for dengue. In order to develop a model for dengue, we need 5 main ingredients that I try to list while giving the short introduction about the infectious disease. The 5 main ingredients are listed here as model parameters. We have the infection rate beta, such as the SIR, we have the recovery rate gamma, and we have the demographic rate U. These 3 parameters here are very well known among dengue community and can be easily extracted from any data you have. However, we end up with 2 parameters here that we have no animal model to tell us what is going on. And we have to estimate from the field data point collected to describe this kind of behavior. One of them I call here Ade Ratio. That would be how much is the impact of individuals in the secondary infection to the force of infection overall. So if we assume that this phi here is larger than 1, means that individuals in the secondary infection will transmit the disease more than individual in a primary infection. But if you assume that this phi is below 1, you would assume that individuals in the secondary infection will transmit the disease less than individuals in a primary infection. The second parameter is also a tricky one, is about this cross immunity period. Remember that I mentioned to you that it is observed in the literature that can last between 3 to 9 months. And that's because the only study existing on that was coming from the Second World War, where the researcher was following the soldiers for the period of 9 months. And they saw that once they get infected, they cannot get infected during this period of time up to 9 months. But this experiment stopped there. And we kept with this information that after 9 months, people are protected, but then they can be infected again. No, as the time passes, we learn more and more about dengue, and some communities reported already this temporary cross immunity being up to 2 years. So we have also to estimate something between 3 to 2 years. And with these 5 main ingredients, we can write down a system of ODEs, assuming different number of strains. So this system, this model can be applied to other infectious diseases. Of course, we have to take into consideration what are the biology behind it. In the case of dengue, it's pretty simple. What we assume here is that the population is divided into 6 groups. We have naive populations, susceptibles, which have never been infected before, the so-called seronegative. And please keep this name in mind because we are going to use it when we deal with the vaccines. These seronegative individuals become infected for the first time with serotype I. And in the case of dengue, the serotype I can be dengue 1, 2, 3, or 4. It recovers from the primary infection from the serotype I. And after a period of temporal cross-immunity alpha, it becomes susceptible again with a pre-existing antibody specific for the primary infection. Okay? This guy here can then become infected for the second time with serotype J. And to be realistic, J must be different from I because we have lifelong immunity against the serotype. Hopefully, so far, so good. And we assume that after secondary infection, the individuals are recovered for 2 reasons. First, we have no data specifically against tertiary and quaternary infection. And second, because of that immunological response I have shown you, we have a very low probability to have individuals infected for the first time in such a level able to transmit, because the immunological response is so fast by activating the pre-existing antibodies that we no longer develop any symptoms. So this is a simple 2-strain type model with 2 infections only in order to try to describe the incidence data we have and that I showed you in the first slides. So far, so good. So in this equation system, I have the susceptible, so the seronegative individuals become infected by meeting individuals in the primary infection rate beta Si, or become infected by individuals in the secondary infection with this factor 5, the Ade ratio, where we can assume to be smaller or larger than 1, when we want to assume that these individuals transmit more or less the disease in comparison to the primary infection. Okay? It becomes infected for the first time, recovers, becomes seropositive after temporary cross-immunity, and get a secondary infection with a different strains after different strain J after seeing a strain I. That's the system. It looks terrible, but it's really simple to understand. And what is that? It's an SIR model with strain structure. Only that. Okay, so this system here, we can evaluate several times. We are interested in the dynamical behavior, so we do not care much about the reality at this moment, but more we are curious on which type of dynamics these models can give to us. So the first thing to look at is the behavior of this two infection and strain model in different ways. We can analyze a minimalistic version where we have no strain structure. So we assume that secondary infection are caused by the same. So we have only one virus, and you have. So let's try to explain this in the state space, in the state flow diagram. So we divide the population into the six classes. We have only one virus. You have seronegative individuals become infected for the first time. And after recovery rate gamma, it becomes recovered from the first infection. And after temporary cross immunity, becomes susceptible again to acquire a secondary infection and recovers. I do not have strain structure, but I have two infections. And I do not assume that the same virus would help the clearance of the secondary infection, but would cause a new infection, as it would be with a different serotype. I have no vector dynamics, but I have to have seasonality because the disease is seasonal. So what I do here is that I put the infection rate beta, I make it as a time-dependent infection rate by just adding a cosine function here. So I have two infection model, primary infection, secondary infection. The transmission rate is time-dependent, and I do not consider more than two infections because I have low frequency of tertiary and quaternary infection. And this gives a system, a very simple system, a five-dimensional system, where I have seronegative, primary infected, become infected with two different infection rates. So that's the force of infection, where I have individuals become infected when meet individuals in the primary infection or when meet individuals in the secondary infection. We have temporary cross immunity, and we have this parameter phi here, which would increase or decrease the transmission rate of individuals in the secondary infection in comparison to the baseline transmission beta for the primary infection. Is that clear? Please let me know if there is something which is not clear, okay? Okay, we can look at the models in terms of bifurcation diagrams. We look at the non-seasonal model, so when we assume that this beta is not time dependent, or the seasonal model, and we see the difference in dynamics. Remember that this one strain epidemiological system is not realistic, is not consistent with what I just told you in terms of biology. We are here interested in the dynamical behavior of the model, okay? So the non-seasonal model gives nothing but a hope bifurcation. You can see here. And I have plots for the parameter alpha, the temporal cross immunity, or the Ade ratio, the parameter phi. I fix phi and I look at alpha, and I fix alpha and I look at phi. What happened? When I fix alpha, when I fix phi and I look at alpha, we see that I have nothing going on. So fixed point, fixed point, fixed point until temporal cross immunity is around 20, and then it bifurcates. Okay, so I would have a periodic solution when considering temporal cross immunity equals 20. That means, so if a year has 52 weeks, alpha equals 20 would be something like 52 would be 1 week. That would be 2 weeks or so. So in order to get this hope bifurcation here, I would need to fix this phi value to 2.6, meaning that individuals in the secondary infection transmit the disease up to 3 times more than individuals in the primary infection, and they are getting reinfected after 2 weeks of clearing up the first infection, which is also not realistic, but that's what we have. When we add seasonality to the system, this hope bifurcation here become chaotic behavior. And I will show you that this is correct. So please trust me, this is chaotic behavior here, deterministic chaotic behavior. We look at the other parameter that we do not know. That's the antibody-dependent enhancement ratio by fixing alpha equals 52. Alpha equals 52 means that individuals are infected. After a week, they are clearing the infection and then they get infected again. Again, not realistic. And we only get anything when fixing the parameter to this point. The same for the phi. If you have phi below 1 or you have alpha below 52, we have only fixed point going on. Okay, so I'm showing only where we can find some dynamical behavior. And here in this scenario, we have also hope verification happening, but only for phi much larger than 1. Similarly, by adding seasonality, this hope verification develops to a more complicated dynamics, which is chaotic as well. We are going to see it. So as we see for the simple SIR with seasonality, we have chaotic behavior by evaluating the Lyapunov exponents, meaning that the largest Lyapunov exponent of the system is positive. We have also for this one, which is a bit more complicated than a SIR, but is an SIR type model without structure of strains. Okay, and then I'm curious how it looks like in terms of time series. When we plot the data in red and you plot the model output on top of the data, for this kind of parameters here, so fixing that temporary cross immunities at least 1 week and individuals in the secondary infection transmit the disease at least 3 times more than individuals in the primary infection, we have irregular outbreaks occurring every 25 years, which is not data-like, all right? So we do not need to do any kind of statistics to reject this model. And we knew since the beginning that we would not be able, I would be surprised if we would be able to describe the dynamics by simplifying so much the number of strains or the temporary cross-immunity or the contribution to the force of infection of individuals in the secondary infection. But at the end, good luck, we cannot describe this data. So we have to add more complexity to the model to see how to get closer to the oscillations we observed in the data. So I'm just getting a look at the data here, the time here. Okay, good. So this system is very rich, although it's not realistic for dengue. And as I mentioned to you, we were able to calculate, for the SIR was the eigenvalues. Here we calculate the Lyapunov exponents. And similar to the eigenvalues behavior, when we have a positive Lyapunov exponent, we have chaotic dynamics. When we have a negative Lyapunov exponent, it contracts towards a fixed point. And when we have a 0 Lyapunov exponent, we have a limit cycle going on. And this can be computed by this expression here. And for this one strain epidemiological system, we can see the 2D Lyapunov exponent showing that you have positive Lyapunov exponent, you have chaotic behavior only, but then only by fixing alpha to be at least 15 and phi much larger than 1. Okay. After that, we have nothing going on besides limit cycle and fixed point here. Okay. This model is not good enough, so we add complexity. And what we do now is we add strain structure. So we now put a bit more of reality, and we assume that individuals in the seronegative individuals can become infected with dengue 1 and dengue 2. Again, we do not have asymmetry between viruses. We do not have asymmetry between serotypes. So we just assume that it's dengue 1 and dengue 2, they are different. If you get a primary infection with dengue 1, you recovered with this dengue 1. And after temporary cross-immunity, we become susceptible again, having experienced the first infection with dengue 1, which means that you can only be infected with dengue 2 after seeing dengue 1. And the other way is also true. When you see dengue 2 first, you only can get a secondary infection with dengue 1 after seeing dengue 2. Okay? That gives us an ODE system with not 6 equations, but 10 equations. So we have a 9-dimensional system. Remember that we assume, similarly to what we have done for the SIR, we assume constant population size. So we end up with a 9-dimensional system. We simplify, we have no vector dynamics, but we have seasonality eventually by adding the cosine function into the infection rate. And the force of infection is explicitly given here. So beta when you get the infection from individuals in the primary infection with dengue 1 or dengue 2, and phi beta when you get the secondary infection by individuals, when we get the infection by individuals in the secondary infection with dengue 1 and dengue 2. Okay, again, two possible infections. Let's look at the equations. We have an SIR model, and here the I can be with strain 1 or strain 2, so that gives the strain structure, differentiating strain 1 from strain 2. And we have an SIR model for the secondary infection. Again, structuring strain 1 and 2, or 2 and 1, depending from whom you get the, which one you get the first infection. Here, you have susceptible individuals become infected by primary infection or by secondary infection with strain 1, and the same with strain 2. And here, after being recovered, you are susceptible again, having seen strain 1 and strain 2, and you can get a secondary infection with a different strain by meeting individuals in a primary infection or in a secondary infection. Please feel free to interrupt if you have any questions, okay? Okay, so we look at the bifurcation diagram by varying the parameters. We do not have any idea. I mean, we have an idea, but we cannot validate because there is no data for that. So the first one is the Ade ratio. We are very interested in knowing how much more individuals in the secondary infection would transmit the disease in compared to the individuals in the secondary infection. And that's because, remember that I told you that we have had most of the primary infections are asymptomatic. So we do not know how many of them are there. We do not know how many of them are there able to transmit the disease. However, when you are in the secondary infection, you tend to develop severe symptoms. So you go to a hospital, you are there isolated. So you are not able to transmit the disease as individuals which are asymptomatic running around. And that was always our issue. What can we learn from the model in terms of contribution to the overall force of infection from primary infection and secondary infection? So this is the bifurcation diagram where we fix the temporary cross immunity to 1 week. Basically, individuals get infected, recovered. It takes a week to recover and then get infected again. And this was proposed back in 1999 by Neil Ferguson. And for the first time, chaotic dynamics was observed for dengue epidemiology only by assuming that individuals in the secondary infection transmit the disease more than individuals in the primary infection. At that time, that made a lot of sense, because if you remember that scheme of the immunological responses, when you get the secondary infection with a different strain, you have an enhancement of viral load. So we have more virus there, and you can assume, okay, these individuals would transmit more than individuals in the primary infection because they have more virus than individuals in the primary infection. Yeah, but the viral load is correlated to symptoms. If you have a lot more of virus, you have more symptoms, you go to hospital, you do not transmit the disease as much as those which have an intermediate level of virus and still can transmit. So what we have done for this model here, we refine it when we add this period of temporary cross immunity to have 6 months interval between the primary infection and the secondary infection. And for our surprise, we found chaotic behavior also in this region where the contribution of secondary infection is smaller than the contribution for the primary infection. We kept this chaotic window here as well, and that was really nice because we no longer needed to restrict individuals in the secondary infection to transmit much more than individuals in the primary infection in order to understand the dynamics of the disease. Okay, so this was a great finding and much more realistic in terms of epidemiology, and because of the hospitalization of severe disease. So we assume that because individuals have more viruses, they go to hospital, they transmit, even though slightly less, but less than individuals in primary infection. Do you want a break or can we continue?
Can you hear me?
Yes.
Yeah, if you want, we can make a break until 15:40. Okay. So about 10 minutes. Yeah.
Okay.
And I posted a question in the chat, so we can discuss it later.
Do you want me to stop sharing and check in the chat?
Well, I think you don't need to stop sharing.
I cannot see. You can read for me then?
Ah, okay, okay, yeah, yeah. So yeah, okay. Or I can ask the question later. Don't worry.
Ah, okay.
Okay.
Okay. So before you go and switch off, because I know that's a lot of information, just to summarize what we have seen so far, we have analyzed an N-structure, an N-strain modeling framework for dengue, where we're analyzing the models by increasing complexity. We have analyzed first a model where we have two infections but no strain structure, and we see that this model is not enough to describe the oscillations on the data. And this model was then refined and open up to a structure level where we have strain 1 and strain 2 playing a role and interact. We look at the temporary cross-immunity and the antibody-dependent enhancement ratio are very important parameters in the model. And by assuming that there is an interval between primary infection and secondary infection, we found a new chaotic window which explained complexities on the dynamics by also assuming that individuals in the primary infection would transmit more the disease than individuals in the secondary infection because of the hospitalizations of the secondary infection individuals. Okay, good. So 10 minutes break and we'll be back soon.
Okay.
3:35, 3:45, I'll be back, right?
Mm-hmm. Yeah.
Okay.
Allá solo con adaptados, allá solo con adaptados. Incluso segundo estrella. Así que tú las funciones no has puesto a ¿sí? Vale, vale. He visto una pasada. Pero vamos a Maíra que no se puede ver. ¿Siete coches? No. Bueno, no sé. No, no sabía, porque son cosas.
Okay.